If I'm not mistaken, we are officially live. Hello, everybody, and welcome to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking the organizers for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world, them being Tim Vogels and Panos Bozellos. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guests from Yale University, Professor Damon Clark. Damon studied physics at Princeton, briefly working on mammalian brain architecture, and during his PhD at Harvard in the lab of Aravi Samuel, he focused on quantitative biophysical analysis of neural activity and behavior in C. elegans. In 2008, he joined as a postdoctoral researcher the lab of Thomas Clandinin at Stanford, where his new interest, visual motion processing, started taking over. With strong contributions in defining the computational structure of motion detectors in Drosophila, he started his own lab at Yale in 2013, if I'm not mistaken, and has remained there ever since, nowadays as an associate professor in the departments of molecular and cellular biology and of physics and neuroscience. Throughout his academic years and in his lab nowadays, they are interested in neural computations as algorithmic solutions to fundamental tasks. Focusing on visuomotor transformations, they employ a wide array of techniques and leverage the accessibility of Drosophila. And today we have the pleasure of hearing about their latest and I'm sure exciting findings in his talk entitled Incorporating Visual Evidence and Counter-Evidence to Estimate Self-Movement. So without any further ado from my side, please all welcome Professor Clark. Damon, the stage is officially all yours.
Great, thank you. It's a real pleasure to be here, and thank you to the organizers for inviting me. Let me get my sharing set up first. And let me see, I have to do one thing here. Even though we went over this before, it still takes time. Okay, here we go. So this you should see the full screen here. We are all set. Okay, perfect.
We're good to go, yeah.
So I'm going to tell you sort of two short stories today. And what I want to do is start by thinking about maybe one of the problems we've all kind of encountered, which is that in neuroscience, there are a really wide range of model organisms that people are working on. And how should we actually go about making comparisons between the results in the labs and the findings in these different organisms? And I think this is sort of a major question across neuroscience. And clearly, I'm not going to answer it today, but I think just to make it a little bit more pointy, you know, animals have different parts lists. So like the molecular components in like flies and primates look totally different. These are two potassium channels. Neuromorphology can look totally different. So, you know, neurons in hippocampus and neurons in the fly visual system just look totally different. They have very different morphologies. And then brain anatomy can be totally different. So how are we to sort of analogize and compare between different animals? And one way to do this is to really think about Marr's levels of understanding. So Marr and Poggio proposed that we should think about algorithms as kind of one level of understanding. And so this is sort of the abstract, like a mathematical abstraction of what a circuit is doing. And then at a level below that, one can think about mechanisms. So this is sort of the molecular biophysics and connectivity and that kind of thing. And then at a level above the algorithm, one can think about performance. So like, why has evolution arrived at this particular solution to the problem? And so what's nice about this framework is that once you've abstracted away the mechanisms and you're thinking about algorithms and performance, you can really start to compare between animals. On the other hand, these aren't super separate. You know, the more you know about one of these levels, the more you can learn about the others. And different animals have all sorts of different advantages in going after the mechanisms. And so I just want to talk briefly about the fruit fly, which I'm going to focus on today, and what advantages it has in terms of going after mechanisms, but also some of these other levels. So in the fruit fly, we have, I would say, three really prime advantages. So one is that we now have the ultrastructure connectome for the entire brain and ventral nerve cord. I guess I update this every year because it used to say half brain and now it says the entire brain, and now I've just added ventral nerve cord. But knowing the connectivity of the brain has really been crucial in trying to dissect how it works. Then there's this whole slew of genetic tools that we have that allow us to manipulate and measure from neurons throughout the fly's brain. And then last, we can connect circuitry to robust, in this case, visual behaviors, but also many other types of behaviors. And so this ability to connect the nervous system to behavior, I think, is really critical for understanding how the brain works. So in my lab, we're interested in how the visual system extracts information from visual scenes and how those signals guide behavior. So I'm gonna sort of start with a short story and then move into a longer story. And the short story I want to start with is from a few years ago, but it's looking at how fly and human motion detection algorithms work and can be directly compared. So I want to start with the idea that motion illusions can actually tell us about visual algorithms. So illusions are typically instances in which our sort of reasoned expectation about what our percept should be is not met. And a really nice example of this is from karaoke. And this is called the snake illusion. And so what you're seeing on your screen right now is actually a PNG, so it's not moving. But what you should see, especially if you move your eyes around and scot around a little bit, is a strong rotation of these circles, sort of relative to one another. And what we wanted to do is sort of take this illusion in humans and ask whether we could find analogous things in flies. So this was sort of how we started out the project. It turns out that the illusion, the rotating snakes illusion, is kind of a complicated version and iteration of an earlier illusion that was studied called the peripheral drift illusion. And so, psychophysics in talks is hard, and it's especially hard when it's over video, but we'll try to do this anyway. So what you should do in order to see this peripheral drift here is not look directly at the center of the circle, but maybe look off to the right-hand side of your screen, but now focus on the circle. And what you should see if you pay attention to the circle is a mild, very mild, clockwise rotation. I'm going to move it to a couple different places and keep focused on the side of your screen, and you may see it move more when it appears in a new place. So if you didn't see it, don't worry. I've got one that will definitely work for you. So now stare directly at the center of this, and I'm going to let it sit there for a few seconds. And now when I remove it, what you should see is very strong counterclockwise rotation when it disappears. And so hopefully that was visible. So this had been studied in humans since the late 70s, but no one really knows how it works. And so what we wanted to do is ask whether flies also experience this illusion. So what we can do is take advantage of the optomotor response in flies, which is a rotational response to rotational movement. So when the world rotates around the fly tends to rotate with it. And so we can use this as a readout for what the fly's percept is. So what we wanted to do is take this illusion, which typically we have sort of rotating like this, and now put it into a geometry where we could elicit rotation from flies. So what we can do is array kind of the outer ring of this around the fly, and then put the fly in a psychophysical sort of chamber here, and present this on screens that are arrayed panoramically around the fly, and then ask how the fly rotates. So what this looks like is roughly like this. So here, this is the little ball that the fly walks on. These are the panoramic screens. It's being shown a different stimulus here, but what we can do is monitor the rotation of the ball and know which way the fly is trying to turn. So it turns out once you made one of these, it's relatively straightforward to make a bunch of them. And so here we actually have 15 of these in lab right now. And this is like one of the real pleasures in lab is that we can ask a question, and about an hour later we can have 15 flies worth of data, and that's usually good to answer a question. And then in the spirit of making comparisons between model organisms, this is my graduate student who built this particular rig, showing that he can't really compete with flies in terms of their behavior here. What we wanted to do then is initially just ask, like, are flies perceiving this illusion? And so we put flies in this rig, and then we presented this stimulus here. So this is this sort of sawtooth stimulus arrayed around the fly. And then we just asked how flies turn in response to this. So we presented it for 5 seconds. We measured the fly's turning rate. And what we saw is during the presentation, the flies turned on average slowly in the direction of sort of the slowly rising derivative. And then when the stimulus disappeared, they turn in the opposite direction. And so this seems to match exactly like what human percepts are. Humans perceive motion again in this direction towards the light side of the sawtooth. And then when it disappears, they counter turn. So here's where I'm going to skip a lot of the details. But using the genetic tools in flies, we were able to sort of develop a, a, a theory for how this arises in flies that we're pretty confident about. And the way this works in flies is that this type of stimulus activates light edge detectors that are sensitive to light edges moving to the right. And it also activates dark edge detectors that are sensitive to dark edges moving to the left. And the light edge detectors have a strong influence on the fly's turning, causing it to turn to the right. And the dark edge detectors have a smaller influence, but which causes the fly to turn in the opposite direction. So we have these two competing effects. And the net result is this mild imbalance. And the mild imbalance causes the flies, the wild type flies, to turn to the light side here. And one of the reasons we know this is that what we can do is we can actually use genetic tools to silence the light edge detectors, which leaves only these dark edge detectors. And when we do that, we find that we can actually invert the percept, right? So now when there's nothing causing the fly to turn to the right, the entire behavior is to the left. And in fact, we see flies turn in the opposite direction. So I think this was like really strong evidence that there's this balance happening between light and dark edge detectors. And when we silence light edge detectors, we can invert the net percept or the net behavior here. So this was, I think, a really exciting thing where we moved then from humans, where very little is known about this, to flies, where we could apply all of these genetic tools and get to this sort of detailed theory of how this works. But then what we wanted to do is then ask, can we take what we learned in flies and actually apply it back to humans? So this is a little hard because in humans, we cannot do genetic silencing, and that was sort of the main tool that we were using in flies. But in humans, you can do something which is sort of the poor man's version of genetic silencing. And that is you can adapt specific populations of neurons in the brain. So for instance, if you present light edges repeatedly, you can adapt those neurons and they turn their gain down. And so this is sort of a way of messing around with human brains that conforms with IRBs and is a reasonable thing to do in lab. And so what we did is we set up a psychophysical test where we could ask how fast humans were perceiving rotation. And we found that they were perceiving it at around like a little bit under half a degree per second. So this is consistent with this very, very slow rotation that you may have seen earlier. Now, what we could do is we could take those same people, and before we present the stimulus, we could present them with these light, like repeated, so 30 seconds of light edges moving back and forth in both directions, and then present the stimulus. And so what this did, and I think this was sort of one of the coolest results we found in this paper, was that we could actually invert on average the human percept to this illusion, right? So on average, people reversed, and then about half of the subjects showed like very strongly inverting percepts. And so what this, I guess the way we interpret this is that the balanced, the theory that we have of this sort of imbalance in the subtraction of these two channels in flies seems also to at least have some resemblance to what's going on in like the massively larger and also very evolutionarily distinct brains of humans. And so I think what we were excited about initially, we were going from sort of humans to flies, but then one of the exciting things here with this was we were able to go sort of back in the other direction and take what we learned in flies and try to apply it to how the human visual system works. Okay, so I want to shift gears here and talk about a more recent work. And really, we're going to go from kind of local motion detection, which is what we were thinking about with this illusory percept, to how animals interpret motion across their visual field. And so to take an example, this is a pretty unusual example of a human flying. So this is a person wearing a wingsuit. This person is using vestibular and mostly, I would argue, probably visual cues in order to navigate uh, through to this very, very small hole in this rock that's coming up here, and then actually make it all the way through. So what's interesting is that we humans have the ability to, this is sort of an extreme example, but we're doing this all the time. We're integrating motion cues in order to navigate our world. And there's a problem though, which is that humans aren't the only animals in the world moving, right? And for all animals, there are stationary objects in the world, and then there are also other creatures moving around. So this is a particularly horrifying example where this lizard needs to escape from what you'll soon see is a valley full of snakes. And these snakes are all moving. And so there are a variety of motion cues on the lizard's retina. So some are from its own self-movement, and some are from the motion of animals around it. And so it's really vital that it distinguish between these two in order to escape, right? So one wants to be able to distinguish between motion that is reflective of your own movement that you can use to interpret like your position in space versus motion that's created by outside agents. And so this is the fundamental question, right? How can you tell if you're moving or if something in the world is moving? And so you've probably had this experience where you've been sitting on a train and you're looking out the window and there's another train right next to you. And suddenly you see that the train begins to move, right? And there's always this question, is it you moving and the other train is stationary? Because sometimes when your train starts moving, the acceleration is very small and you can't really feel it. Or has the other train started moving and you're in fact stationary? So this is actually difficult if all you can see is the train. But if I give you a slightly larger scene, you'll be able to make this inference perfectly. So let's give you this scene instead. So if what you see out your window is this, where the train and these sort of telephone poles in the back are all moving. So if the whole world is moving, then it's your own self-movement, right? If, on the other hand, you see something like this, where there's some things that are still stationary and some things that are moving, then it's not you moving. So this is sort of the fundamental logical step that we're going to investigate here. And so the point is, if you're a human or a fly, when you are rotating yourself, so this is sort of self-movement, what you see is the entire world rotating around you, right? On the other hand, when there's a bunch of stuff in the world that's rotating, they're often going to be that's moving around you. So there's motion in the world around you or movement in the world around you. There are often going to be objects in the world that are not moving, that are stationary. And so can flies exploit these sorts of geometrical cues by sensing stationary patterns? In other words, can they recognize can they use this sort of strategy to recognize whether what they're seeing is caused by movement of the world or is caused by their own self-movement? So we're going to use sort of the same strategy I talked about in the prior example. So we're going to pin flies in place above a little air suspended ball. We're going to present visual stimuli on panoramic screens around the fly, and then we're going to measure the fly's rotation. Now we should talk a little bit more about what this optomotor response is, how we actually interpret it. So flies turn in the direction of visual motion in a way that stabilizes their orientation, right? So what you can imagine is for, let's say, a fly flying along, if it gets bumped off course to the right by the wind, so if it turns to the right, then it will see motion to the left across its visual field. And if it engages this optomotor turning response, then it will turn to the left, and then that serves to stabilize its trajectory in the world. That stabilizes the orientation. So this is sort of a negative feedback behavior that is thought to stabilize orientation. And so what we wanted to do is ask, so if flies perceive, if they believe that their visual stimulus is consistent with their own self-movement, then they should turn in order to stabilize themselves. But if their percept is that this is not their own self-movement, this is something else in the world, or something outside in the world that's moving, then this optomotor response should not be engaged, right? So that's kind of the logic that we're going to use here. So how do we actually do this? So what we want to do is ask, do flies sense stationary patterns? And so what we can have then is we can have this virtual cylinder, which is outside of these screens, and then we project it onto these 3 panoramic screens around the fly. And then what we're going to do is put a very particular designed pattern or movie onto this cylinder. So what does that look like? So the kinds of movies we're going to look at have we call these sort of islands of motion stimuli. So there's a foreground, which has some pattern on it. And so the 3 patterns we're going to focus on today are a uniform gray pattern, a stationary high contrast pattern, or a flickery high contrast pattern. So these are the 3 patterns that are in the foreground. And then the background is a random binary checkerboard pattern, which just moves in one direction. But you can't see it where the foreground is presented. You can only see it through these sort of little holes that we have in the foreground. So it looks like kind of islands of motion in a sea of whatever the foreground is. So what does this actually look like? So these are the 3 stimuli, and so this is the uniform foreground, right? So this is what you would imagine is a very standard visual stimulus we might use in lab. So the whole screen is gray, and then you've got these like little regions that are that all contain motion to the right. In the next movie, we have a stationary foreground, and so this is exactly the same motion cue, right? So the islands in this particular example, these islands are identical, and we now, instead of having a uniform gray foreground, we now have this high contrast random binary checkerboard pattern. And then in this last movie, we have flicker. So now at 15 hertz, we just update this random binary pattern and cause it, instead of being stationary, to be sort of dynamic and changing. So one of the key points here, though, is that all 3 of these stimuli have the identical net motion energy, right? So when you calculate the motion energy here, these all contain the same amount of motion to the right. And so what we wanted to do then is ask like how flies respond to these. But let's go over just quickly about like what we're inferring here. So the uniform gray provides no evidence of motion. In other words, if this gray moves back and forth, we cannot perceive motion from inside the gray. So this works better if I put an aperture over this. So you have to trust me, I have this thing moving back and forth behind this, but you can't tell, right? Because this gray provides no information about the movement of the pattern. On the other hand, if we take this random binary high contrast pattern and we slide this back and forth, now there is evidence about whether this pattern is moving or not moving. So in other words, now I'm going to put the same aperture on it. I'm going to slide it back and forth. Now you can perfectly well judge, now it's not moving, before it was moving. So instead of providing no evidence of motion, this now provides evidence of no motion. And so what we found when we presented these 3 different stimuli was that flies turn in the direction of the motion when we have the uniform foreground. So this is, reasonably strong turning. When we have a flickery foreground, flies suppress their turning. And I should say this is a sort of known result from an earlier paper in the field that this should happen. But then when we present this stationary pattern, we see this very, very strong suppression of turning. And so when we quantify this just by averaging the turning over the period of the presentation, we find sort of an 80 to 90% reduction in the fly's turning by the existence of this stationary pattern. And so the way we interpret this is that what the fly is doing in order to guide its turning is it's making an inference about whether the motion cues it is seeing are due to its own movement or due to the movement of something in the outside world. And if it's due to something in the outside world, it shouldn't turn, right? There's no reason to turn. But if it's due to its own self-movement, then it should turn to try to correct this deviation. This is like the negative feedback. So we now want to investigate and see whether this is sort of a sensible interpretation of what's going on. So first we wanted to ask, how fast does this suppression occur? So here what we could do is we could present like just the foreground, but with the background stationary. Then we switch, we start the background moving, and then we go back to a stationary background. And so this looks very much like there's a weird binary world sitting there. Some parts of it start moving, and then it goes back to stationary. And so what we saw then was a very similar pattern to what we saw earlier. So these are half-second. This is like a half-second pulse of background motion. And when the foreground is uniform, the flies turn strongly. And when it's stationary, they suppress their turning. But what we'd like to do now is really zoom in on the dynamics of this, right? So what does this look like in the early time? And what we can do is plot here the difference between the uniform and stationary. So this is basically taking the difference for each fly between this gray line and this blue line. And so we can see this start to rise around 100 milliseconds from motion onset. And then if we actually calculate the probability that these are from two different distributions, we see that p-value becomes significant with this arbitrary 0.05 cutoff, like right around 100 milliseconds as well. So what this means, though, is that the flies actually, this 100 milliseconds is pretty similar to it's comparable to the time on which the behavior starts, period, right? So this isn't some suppression that's taking a while to kick in. This is suppression that's sort of immediate from the outset of the behavior. So what we've talked about so far is rotation, right? And there the argument was like, if you're rotating, then everything in your field of view is also rotating. But this isn't true of all self-movement. And in fact, for translational movement, it is absolutely not true, right? So you can imagine if you or a fly are just walking through the world and there's, a very distant object like a mountain or the sun or the moon, and you are just walking in a straight line, that object is always at exactly the same like azimuth and elevation relative to you, right? It is stationary, even though you are moving through the world, right? So for translational movement, the existence of stationary objects does not imply that you are not moving, right? For translational movement, you could well be moving and you could have these stationary objects out in the world. And so what we wanted to do is test whether this was true in flies. So we know that flies regulate their walking speed based on visual cues and visual movement in particular. And so what we did is we set up the screen so that it would create translation-like optic flows. So instead of rotating around the fly like this, we have the flow sort of emanating from the front of the fly and then passing it this way, or going from behind the fly and moving towards the front of the fly. And so when we present the fly now with this sort of stimulus, we can now ask, like, how much does the fly pay attention to these different stimuli, right? And what we find is that when the fly is when the motion is presented from front to back, so this is like forward, we see that the stationary and flicker stimulus both suppress walking speed a little bit compared to the uniform. But when there's back to front, there's no difference between the uniform, stationary, and flicker. And so the way we interpret this is that stationary and flicker are kind of looking the same here. So the fly is not making any additional inference based on the existence of these stationary cues relative to flicker cues. So we think this is consistent with the idea that the fly is not strongly regulating its walking speed dependent on stationary cues. So now we want to ask whether the structure of the stationary pattern matters. And so you can imagine that horizontal patterns provide no information about horizontal motion. So I can move this back and forth. When I cover it with an aperture, you can't tell whether it's moving or not, right? On the other hand, If we have vertical patterns, these do provide information about horizontal motion or absence of motion, right? So when this moves back and forth and I cover it with the aperture, you can clearly distinguish the motion from the absence of motion here, right? And so what this means is that these different orientations of contrast provide different kinds of evidence to the fly about motion in like its yaw, in the yaw plane. And so what we could do is we could make three different foregrounds. So we have the three different foregrounds, one of which has strong vertical bars that are, again, random black and white bars, one of which is 2D, so this was the same as the original, and one of which has horizontal bars, right? And so we can make a strong prediction, which is if the fly is indeed using these patterns to make inferences about its own self-movement, then stationary vertical patterns are much more informative about the fact that it is not rotating than horizontal patterns are. Horizontal patterns are not very informative about that. And so we would expect the suppression to be strongest for vertical patterns and weakest for horizontal patterns. And I guess this is really a control to test whether there's sort of a generic suppression of turning based on high contrast stimuli, or whether the structure of the stimuli also matters. And so what we found was that the horizontal stationary foreground caused a little bit of suppression relative to the uniform foreground, but that when we had the vertical pattern, we saw the strongest suppression. And so we could quantify this. And so again, we're getting very strong suppression by those cues, including the 2D cue, that have information about motion in the horizontal plane. So what is the fly doing here? It has sort of an algorithm. And for specific behaviors, so rotation, flies are distinguishing between two types of zero velocity visual cues. In one type of cue, there's the absence of evidence, right? So this is the gray foreground, contains no information about movement, right? And then the high contrast stationary pattern also has zero velocity, but it actually provides evidence of the absence of motion. So the fly is really distinguishing between these two types. So let's think about like what the mechanism is for this in flies. So this is one of the advantages working in flies. We can go through and we know a lot about the visual system and have a lot of tools to investigate this. So I want to go over first like directional circuits in Drosophila, and this is based on the work of many labs. So guiding the fly's rotation and also translation is this visual circuit here. And so if we start out looking at the sort of the photoreceptors here, they are behind these facets on the fly's eye. You know, Gary Larson has this old cartoon which shows what a fly might see right before its imminent death. But this is actually one of the few cases where Gary Larson got this totally wrong. This is not at all what the fly sees. In fact, what a fly would see through these many facets is not an image for each facet, but basically a pixel for each facet. So this is what the fly would see. So each facet you can think of as like a pixel in its visual field. And then what we can do is sort of expand and go take a cut through the fly's head. This is like that section of the fly's brain. And then over here, so these are the facets on the outside of the fly's eye. And then this here that I'm highlighting is the fly's visual system. And so this is where we spend most of our time focused. Now, at sort of a few synapses in the visual system, there are these two neurons, T4 and T5. And T4 responds selectively to moving light edges, and T5 responds selectively to moving dark edges. And so if you're a vertebrate, if you're a person who studies vertebrate retina, this should look very familiar. These look a lot like starburst amacrine cells, the on and off starburst amacrine cells that have been studied in mouse retina, which in the mouse retina are the earliest direction selective cells. So it turns out there are 4 T4s and 4 T5s that are arranged in this sort of cruciform pattern of selectivity, so that we have one selected for upward, one downward, one leftward, one rightward motion. Then we have one T4 and one T5 for each of those 4. These then tile all of visual space. So there are 8 of them per facet in the fly. And so then you can think of them as like tiling all of visual space. So this is sort of where this is the substrate we're trying to work with in order to figure out um, how flies are uh, detecting and using these stationary cues to modulate their behavior. So we should step back, and a lot is known about how animals seem to integrate motion signal to detect self-motion. And in particular, in flies, zebrafish, primates, um, it's thought that, or there's a lot of evidence that these animals use an optic flow template. And so what this template looks like is something like this. So this would be essentially the flow that would be elicited by a particular movement of your body. In this case, this would be the flow elicited by pitching forward. So there would be like motion up directly ahead of the animal, motion down behind the animal, and then this sort of swirl on each eye as this happens. And so what the way these sorts of models work is that one creates a flow template for all sorts of different rotational axes. And so here we just have a pitch axis, a roll axis, and a yaw axis, but you could imagine other sort of non-canonical axes of rotation here. And then you have the animal's measured optic flow, right? And so this is the output. In the fly, this would be the output of like the T4 and T5 neurons at all these different locations in space. And then the way this is basically thought to work, and this there's sort of a straightforward way to do this with neurons, right? Is you can take, sorry, see if this works. You can take this measured optic flow, and you can basically just take the dot product of the measured optic flow with each of these templates, right? And so this gives you a single number, which is how similar your current flow is to what you would expect from pitch, right? And then how similar is it to what you'd expect from roll? And then how similar is it to what you'd expect from yaw, right? And so then you get these three numbers out in this case, and you can use those to determine your self motion. So this is the idea, and there's a lot of evidence that for neurons that have these sorts of receptive fields that look like they're taking these sorts of dot products. However, what we've shown just now is that this is not sufficient to explain the fly's behavior, right? Because all of this sort of white part here in the measured optic flow is just a zero velocity signal. But what we've shown is that there are two types of zero velocity signals, right? There's a zero velocity signal that is elicited by a gray stimulus, which means that there's no evidence of motion one way or the other. So you take it or leave it. There could be motion, there could not be motion. We just can't tell, right? So that's one kind of zero signal. And then there's another kind of zero signal, which is that there's evidence of no motion, right? And so this is a high contrast stationary pattern. Now you know that there is zero motion, right? It's not that you can't tell whether or not there's motion. There is actually zero motion. And so these two are clearly, our evidence shows, treated very, very differently by the fly as it's deciding whether to turn. And so what this means is that the template matching can't really explain our results. The template matching is clearly happening. So we want to know, like, how is this going on? What's happening here? And so we developed sort of a simple model for how you could create sort of suppression by stationary patterns. And that's basically to have a parallel stationary pattern detector. So this is sort of the canonical motion detection and behavioral circuit in the fly. And now what could happen is if you had stationary pattern detectors, you could imagine them suppressing motion detectors, so suppressing the activity of the local motion detectors, of the optic flow detectors, or of premotor descending neurons. So these ones are like looking at little local motion detectors. These optic flow detectors are basically these templates. And then downstream from those, we have putative premotor and descending neurons. In the fly, some of those are known, but many are not. So what we wanted to do is look for neural correlates of this behavior. And so in particular, we wanted to start off by doing some two-photon imaging where we can show the fly very similar movies, or actually identical movies to what we showed them in behavior on panoramic screens about them under a microscope, and then measure from these neurons in the fly's visual system. So these are the T4 and T5 neurons. So these are the earliest motion detection like local motion detectors in the fly's visual system, and then these what are called lobule plate tangential cells, which are thought to be these optic flow measurement neurons that do kind of the template matching. And so we can just express GCamp in these neuron types and now measure the responses we see. And so here's what we see with T4 and T5. So what we see is sort of relatively strong responses to these stimuli when we have a uniform foreground, and strong suppression when we have a flicker foreground. And again, this is a known spatial contrast adaptation that was known earlier in flies. But what we see is that the stationary patterns are not suppressing T4 at all and are just a little bit suppressing T5, right? So this is not at all the pattern that we saw in the behavior. And so we think that this is not what's happening. So stationary patterns are not suppressing the activity of the local motion detectors directly. So what we can do is we can take our simple model and we can update it, sort of cross this off. We see no evidence for stationary patterns influencing like the local motion detectors. So now what about these optic flow detectors? So we can record from those instead. And there are a bunch of specific drivers. This is by no means all of the LPTCs in the fly's visual system, but we can record from these. And what we see is basically no patterns that look like the behavior, right? So it's very hard to sort of look at these patterns and see a case where we have like suppression by stationary and then not much suppression by flicker, right? So we interpret this to mean that there's basically we can't see any evidence here for anything analogous to the stationary pattern detectors like directly inhibiting optic flow detectors. So we sort of cross this off of our model. And now what we wanted to do is look for evidence of these stationary pattern detectors like influencing the sort of downstream of the optic flow detectors. So do we have evidence for this? And so what we decided to do is an experiment where we could look for stationary detection transferring between the eyes. So we could present one stimulus on one eye, a different stimulus on the other. And so on each eye, we're either going to present uniform gray screen, stationary high contrast pattern, the flicker pattern, or full field motion. So if there were local suppression, then that would be happening kind of in the optic lobe. And so then presenting stationary patterns on the other eye. So this is now presenting motion on one eye and gray on the other versus motion on one eye and a high contrast stationary pattern on the other. We wouldn't expect these to interact if it was all happening locally. If, on the other hand, you had global suppression, this was happening in the central brain, then presenting motion on one eye would be affected by stationary patterns on the other. So the stationary patterns here would suppress the turning. And so it turns out this is exactly what we see. So let me show you the data here. So what we can do is present back to front motion on one eye and gray on the other. So we present this on the left eye and gray on the right. And then we can also present gray on the left eye and high contrast to the right. And then what we do is we take a linear sum of the responses to these two stimuli, and we compare it to the response to the conjunction of these two stimuli. And what we find is when these are presented in conjunction, we see a very strong suppression of the turning response when the stationary stimuli is presented on one eye and the motion is presented on the other. And so this leads us to believe that there's some central mechanism in the central brain of the fly, not locally in the visual systems, that's causing this suppression of the optomotor turning. So then we wanted to take advantage of what we can do in flies to screen for the neurons involved. So in flies, we have a lot of fairly specific drivers, and then we developed this high throughput behavioral psychophysics rig, which allows us to actually do kind of a targeted screen of different classes of neurons in the fly's visual system and some in the central brain. So we're going to call this a moderate throughput screen. And this is one of these slides where it'll take a minute to present, but it took a year to get. This is like 1,200 flies worth of data here. What we're going to do is silence these neurons one at a time. And so we did that with a tool called Shabiri TS. And then we're going to compare it to a genetic control. And we're going to ask what was the fractional turning in response to the foreground, the high contrast foreground relative to the gray foreground. And so no suppression would be 1, and strong suppression would be 0. And so what we can see is, so we went through a bunch of neurons in the early visual system. We went through a bunch of neurons in sort of the intermediate visual system, one synapse down from those early ones. We went through a bunch of other DMs, which are kind of local inhibitory amacrine cells in the fly's visual system. And then we went through a few in the fly's central brain that are known to govern orientation. So we then looked for hits among these, following which we retested those flies that had hits, and found some got knocked out on retesting, but some remained. And so we ended up liking this driver the best. This was the one that we focused on because this was a very clean driver. It hit a single neuron in the entire brain and had this strong silencing phenotype that led to less suppression of turning. These other ones were also exciting, but tended to hit many neurons throughout the fly's brain, not just a single one. And so it was harder to go after the neurons with these particular drivers. So the neuron that we identified there is called MI4. And this is a stain from flylight of what it looks like. And so this is an early visual neuron that happens to provide input to T4, but also synapses onto various other neurons, about which very little is known. So we wanted to measure now the responses of MI4 to these stimuli. So the first thing we did is we presented stationary stimuli and flicker stimuli to MI4 and measured the response. So this is kind of the baseline MI4 response here. We presented this for 2 seconds, and we found that these looked kind of disappointingly similar. MI4 is responding not very much to either stationary or flicker stimuli. But now what we could do is we'd actually recorded from these across the visual system. And so some were seeing like light regions on the screen, some were seeing dark regions, and these MI4 cells are known to be on cells. And so when we separated out our responses by percentile, what we found was that there was a very large variability in how strongly MI4 was responding to stationary stimuli, and a much smaller one in response to flicker. And this is actually very sensible considering what else is known about MI4, which is that it seems to be a slowish integrating cell. So it tends to integrate in time. And so flicker would tend to just be averaged out, but a stationary stimulus would not be averaged out and would elicit a strong deviation from baseline. And so that's exactly what we saw here. And note also that sort of unusually, I would guess in the fly visual system, in MI4, we can see hyperpolarizations with our calcium signal. So there's a sort of relatively high baseline calcium, which can decrease. And that's a little unusual. There are many cells in which you cannot see this sort of effect. So we wanted to just develop a model for how a stationarity detector would work using MI4 inputs. So MI4 is inhibitory, and we suppose that there's a local neuron X downstream of it, followed by a stationary pattern detector downstream of that. So this is just what we want to go after. And what we're going to do is have both the MI4 and neuron X have some constitutive activity, which can then be modulated. So MI4 is inhibitory. So when MI4 is presented with uniform, or when this model of MI4 is presented with uniform or flicker stimuli, it doesn't deviate much from its baseline, which means it continues to inhibit neuron X. So neuron X is never active. So that's sort of one thing that's happening. On the other hand, when there's like a strong stationary pattern that's long-lived, some MI4 neurons will be activated, in which case they will continue to inhibit their postsynaptic partners, but some will actually be hyperpolarized, especially those looking, for instance, at like strong black stimuli, right, that are there, that are persistent in time. And in those cases, the postsynaptic part is disinhibited and becomes active. And so now if you integrate over those stationary patterns, over those neurons X, you can develop a pretty nice stationarity detector, right? So this is just a simple proof of principle that these MI4 signals can be used as the input to a stationarity detector that works quite well to detect stationary high contrast patterns, but is not strongly activated by either motion or flicker. And so what we think then, after this work, is that there's the sort of classical motion detection pathway in flies that is known to drive optomotor behavior. And then that there's a parallel pathway that seems not to interact much with the motion detectors in the visual system that leads to the stationarity detection, which influences turning deep in the central brain, and has a neuron, MI4, that is appears to be involved early on in that pathway. Clearly, there could be other neurons, and this is sort of this model is a sort of sufficiency model to show that MI4 could do this rather than any sort of real bet that this is what's happening. Okay, so to conclude, what we found in sort of the second part of the talk that I focused on is that flies are, when they're turning in response to visual motion, they're making an inference about whether the motion is due to their own self-movement or due to movement of objects in the world around them. And when they make this decision to turn in response to these visual stimuli, they're integrating evidence both for and against the hypothesis that the visual stimulus is created by their own self-movement. And in doing this, flies are actually distinguishing between sort of these two types of zero motion signals, one of which is this sort of gray, which has no evidence about motion, either for or against, and another of which has evidence of the absence of motion. So that's these high contrast stimuli. And so this distinction that flies are making is really not compatible with any of the match template models, because those treat zero motion as a zero vector, and it's not qualitatively different when it is arising from seeing gray or seeing something stationary. So stationary patterns are detected in parallel to early motion detectors. So we don't see influence of these stationary patterns on the signals in the early motion detectors or in the optic flow detectors that we measured from. And then I guess the last point here gets back to where we started, which is the arguments that are being made here about why stationary patterns potentially should suppress this optomotor behavior are based on really simple geometrical principles about what the world looks like when an observer moves versus doesn't move, and when objects in the world move versus don't move. And so it seems very likely that these sorts of phenomena will be widespread across animals that use vision to stabilize their movement through the world. Okay, so I want to finish just by thanking my lab, and in particular the lab members who contributed to the work that I presented here. So the early work on illusions was done primarily by Margarita and Ryosuke, and the subsequent work on stationarity was done by predominantly by Ryosuke, with a lot of help from Baohua, who worked on some computational work that I didn't have time to talk about, along with Braden, Natalia, and Bara. So with that, I would like to finish and take any questions. Thank you.
Thank you very much, Damon, for this fantastic talk. I can't help, but I need to start with saying how jealous I am of all the tools and increasing accessibility you have in dissecting, the very specific contributions of single cells in these fundamental computations. I went ahead and I posted the Zoom room link in case people want to start joining us already. You can also post your questions in the chat. And there was a funny comment from Wei Li for the first part of your talk when you showed the member of your lab trying to recapitulate the Drosophila behavior, saying that there is a limitation. The subjects were not head fixed. Obviously, joking. So yeah, as we are waiting for questions from people, let me start with some. Maybe one naive one is, I'm like I don't know if this is known, but I noticed that there is a difference in the onset and offset behaviorally when it comes to the latency of responding to, the appearance and disappearance of background motion. Is this of interest or not really? I think it was like in the first figures that you showed.
Oh, in the illusion, or in the first part of the talk, or in the second part?
In the second part, like right
So,
after the, yeah.
yeah, so there's, I guess yeah. So in lab we measure the turning in response to the rotational motion, and then when it stops, you're saying sometimes it appears to go straight down, sometimes it sort of has a longer tail, and sometimes it
Mhm.
actually flies can turn in the opposite direction. We we see all of these, and it doesn't appear to be particularly consistent over time and over experiments. So we've had a hard time interpreting what happens when the stimulus disappears. I think it's a super interesting question, but because we've had a lot of variability in it, we've essentially chosen not to study it.
For
But I
future work.
think but
Yeah.
I think it is it's an important question about the dynamics of this behavior. And I think uh, but it also gets into questions of like how much does the inertia of the ball affect the fly's behavior, and some other things that I think are uh, important but slightly tangential to some of the like visual computations that we're most interested in.
Right, thank you. The first question that appears in the chat is from Aristides Arenberg. Have you tried to use single moving patches instead of multiple moving patches in the view field? Because having multiple patches moving in the same direction might be less natural.
Yeah, so it's a good question, like how to do this. So there were a couple unnatural things. One is actually that the motion, the patch of motion stayed stationary relative to the fly, which is very unusual, right? Usually when something's moving to the right at one point, at the next point it is to the right of where it was, and so it would move across the field. It turns out um, that doesn't seem to make a big difference. The question of density of like these moving patches is an interesting one. And we have tried varying the density of those patches. And I think as you go to smaller and smaller patches of movement, the behavior becomes less and less. It still gets suppressed uh, by these stationary objects. I guess it's yeah. So but part of this is a question of just pragmatically how to design experiments so that you see a large enough signal. It is true that one might not always expect to see like large patches of movement, but if you think about like grass waving in a field when it's windy, like large patches of the grass can all be moving. Trees can similarly do this. So I guess it's not totally clear to me, and I guess this is an interesting broad question of like how should we think about what the actual statistics of this sort of like world movement are compared to like the statistics of the animal movement?
Thank you very much for addressing this. Given that Ari is here with us, maybe we can continue later on offline if he wants to ask a follow-up. Two more questions, if I'm not mistaken, appear from Alon Polek-Polski. Were the responses in T4 and T5 different in lines without the MI4 activity? And the second question, was flies' behavioral responses different in other tasks?
Yeah, so with the so T4, so MI4 is inhibitory into T4. And I guess not experiments from us, but experiments from other labs have shown that silencing T4 seems to affect, silencing MI4 seems to affect T4 responses to motion. So I think that would, I think very clearly, we would expect to replicate that. And so we did not actually do the experiment where we silenced MI4 and then measured for these differences. But I think the real question we were trying to ask was whether whether T4 and T5 were even being suppressed by stationary stimuli, and we did not find that. So silencing MI4 is sort of it's sort of logically one step beyond that. And then, sorry, what was the
The
second
second
question?
one was if flies' behavioral responses
Oh, yeah,
are
yeah.
different in other tasks.
Yeah, so this is an interesting question, because we looked at this trend, like walking slowing task. So I showed you that briefly, where we didn't see the same kind of strong suppression by of the behavior. So the behavior is that flies, when they see motion, slow. And so what you might expect if you had the same kind of suppression for translation as rotation as you would have expected to see the stationary stimulus like not causing the slowing, right? And that is absolutely not what we saw. We saw that it was comparable to the other stimuli, including to the uniform background, or stronger than the uniform background. So that's that was one behavior we tested. We also tested sort of a behavioral response to like a fast-moving bar around the fly, and there we didn't see any differences when there was a stationary background versus a gray background. And so this again leads us to believe that this is a very specific response to sort of broader visual motion rather than kind of local motion that doesn't seem to affect motion detection generally. It seems to be very specific to motion detection as used to drive the specific rotational response in the fly.
Right. Thank you very much for addressing this as well. And given that there are no more questions appearing in the chat, if it remains like this after my last question, we will continue offline with people that are interested. So I'm going ahead and posting the Zoom room link again. So one question I have is, like, probably it could be applied to any part of your talk, but with specific focus on the two stimuli of zero velocity, right? The gray background and the structured background. I was wondering what role the saccades might play in this, because like Lisa Feng, I think with Gabi Maimon showed that there are saccades in flies.
Mm-hmm.
And if you saccade on a gray background, essentially nothing changes, right? Whether when you have structure and you saccade, a lot of stuff changes for individual neurons. So I was wondering what role saccades could play in all these behaviors that you study.
Yeah, I think the role of saccades in fly optomotor turning is not yet particularly well understood. It's a really exciting sort of new finding. And I think that's absolutely true. So I think what one might expect is that behaviors, including saccades, or including, I mean, so the photoreceptors in flies like can both do kind of slow tracking as well as sort of more saccadic like movement. And so one could imagine them also being used to stabilize scenes and that kind of thing. And so one might expect stabilizing behaviors beyond optomotor turning, including these sort of slow photoreceptor movements, to sort of have a similar set of properties associated with them, where they would actually be suppressed by the presence of stationary patterns. Like what the interaction is of the saccades with optomotor turning, I think is like a really not well known yet in flies, though I'm sure the people who are going after it are going to do a great job there. So I'm excited to see what they find.
Right. Thank you very much. And given that there are no more questions in the chat, I think we can also wrap it up and continue offline. So thank you very much once again, Damon, for this fantastic talk.
Great. Thank you for having me.